Hi, everyone, and welcome to People Behind the Science. I'm your host, Dr. Marie McNeely, and I am glad to have you here for episode 744, featuring our guest, Dr. Ryan Potts. If you like this episode, listeners, and you want to check out more stories from amazing people doing great science today, you can find more episodes of our show on our website, peoplebehindthescience.com, Apple Podcasts, Spotify, or wherever else you like to listen. And today, Ryan has some fantastic stories of his own to share with us. So get ready to meet another one of our brilliant people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am excited to be speaking with our guest scientist, Dr. Ryan Potts. So, Ryan, welcome to our show today. How are you?
I'm doing very well. Nice to connect with you and look forward to discussion today.
Well, we are so excited to have you with us, Ryan, and we're looking forward to learning more about you and your work today. And listeners, to give you a little bit of background, Ryan is VP of Research and head of the Induced Proximity Platform at Amgen. This platform works on ways to bring two or more molecules in close proximity to each other to tackle drug targets that are currently considered undruggable. He also leads Amgen's Research and Development Postdoctoral Fellows Program. Ryan received his bachelor's degree in biology from the University of North Carolina at Chapel Hill, and he was awarded his PhD in cellular and molecular biology from UT Southwestern Medical Center. After completing his PhD, Ryan served on the faculty at UT Southwestern Medical Center for 8 years before accepting a position on the faculty at St. Jude Children's Research Hospital. He joined the team at Amgen in 2020, and in our interview today, Ryan is going to share more about his science, of course, but also more about his life outside of science. So, Ryan, can you first start by telling us what you do like to do when you're not doing science?
Oh, well, outside of science, I get into a lot of different things. Obviously, I spend a lot of time with the family and the kids and all that kind of stuff. That seems to take a lot of my time these days. But beyond that, we like to do a lot of things in nature, hiking, etc. We really enjoy the Santa Monica Mountains here. It's a great place here in Southern California to explore the beaches, etc. We really like to travel as well. I like to travel. So we really enjoyed that coming out of the pandemic, both in the U.S. as well as internationally. Then the final thing I'll note probably is I'm a big sports fan. I grew up around sports and continue to really enjoy basically any type of competition, to be honest.
Fantastic. Do you have a favorite sport that you like to play these days?
Well, I guess my favorite sport to play is probably with the kids still is basketball and soccer, but I'm also not necessarily playing, but a big sports fan in terms of football.
Oh, that's awesome. Well, it sounds like, Ryan, you've got a diverse array of hobbies to keep you busy, and I love to hear that. I think so many of the scientists that I talk to are not just all about the science. They do like spending time with family, traveling, sports, all these different sort of facets of their lives.
Yeah.
So let's talk about the scientific side. I know you're doing some really cool things in science as well. So Ryan, how do you describe what you do to someone who's not in science or maybe not in your specific field?
Yeah, thanks for that question. Obviously, this is what gets me excited is to talk about what we're doing in our research labs. And, at a really high level, it's scientific research that's focused on really early drug discovery, where we aim to make impactful medicines to serious diseases with high unmet need. And we work in sort of the earliest phases of that process, doing discovery around biological pathways and targets, disease drivers, and then figuring out how to actually drug them. It's an amazing process to go through, a really challenging one. You have to use lots of creative, innovative approaches to do such.
Absolutely. I think this field of drug discovery is a really exciting and really motivating field to be in. And as you alluded to, of course, there are challenges, and we love helping our listeners get through some of the challenges they may be facing in their own lives. So we love talking about motivation and inspiration and sharing some of the things that motivate scientists. So Ryan, do you have a favorite quote or a saying, or maybe just something in general in life or science that keeps you motivated and inspired?
The thing that comes to mind in terms of motivation is something that when I was starting my career and just training as a PhD student, the very first day of that, which, of course was like chaotic and, just thrown into the deep end, right? Coming into graduate school from your days in sort of undergraduate and sort of working in the lab, et cetera, now into your PhD program. I did my PhD at UT Southwestern, and there's just an amazing set of faculty and mentor there. And on day one, they had all students attend a lecture by one of the most notable scientists there, Joe Goldstein. He's an amazing scientist that discovered sort of the cholesterol biosynthesis pathway and won the Nobel Prize with Mike Brown, who was also at UT Southwestern. But Joe came and talked to the students on day one to sort of inspire and motivate folks. And I think one of the things that he emphasized was sort of a real classical quote, but I think has a lot of applications to scientific research. And I think it's from a Roman philosopher, but it's sort of this concept of luck is what happens when preparation meets opportunity. And I think that has a lot of parallels to scientific research. I think a lot of people may misinterpret like being lucky, but I think it's really more that luck finds those that are prepared. And that stuck with me from that point on is, you have to be on the lookout for observations that are going to be important and impactful. And if you're prepared for those, you'll find them. If you're not, then you'll sort of miss potentially transformative discoveries.
Absolutely. And for you, Ryan, were you always one of these sort of ultra prepared people, or was this a skill that you sort of had to learn along the way?
No, I think I'm probably more in the prepared situation. I'm more kind of person that, would always sort of write out the protocols for what I was doing in the lab or, have a schedule of when I need to do certain things to meet deadlines or et cetera. So I'm usually not the kind of person that just flies by the wind on these things. I'm usually pretty prepared.
I'm the same way myself. Very type A, I would say.
Yeah.
Well, you mentioned Joe Goldstein, and I think this is a huge figure in the field and certainly an inspirational one. So when you think about people along your journey who may have impacted you, and perhaps Joe is one of them, are there other mentors or role models or people who just really rise to the top of your mind as those who inspired you and had a big impact?
So many different role models for me, I feel like, that really shaped who I was as a scientist and probably a person. I can just maybe name a couple, but I want to start with maybe my first real mentor, and that was Mohanish Deshmukh. And he was just starting a lab directly out of postdoc. And this was at the University of North Carolina at Chapel Hill. And I had just finished undergraduate there and was looking to work in the lab as a research technician for a little bit before deciding whether to go to graduate school or not. And I got the opportunity to join his group basically at the same time that he started. So I was like employee one, and there was nothing in the lab. Like literally I walked in and I was like, this is like an empty room. And I was like, okay, that's the first challenge is we had to get through is building the lab. But because I was sort of the first person that joined that lab, I got a lot of attention. And Mohanish was a great mentor to me to sort of learn how to do science in the right way. And this second person I'd highlight is a little bit later in my career path, and that was Steve McKnight. Steve McKnight is an amazing scientist and made many notable discoveries. He's at UT Southwestern. He was the chair of the biochemistry department that I was in. The thing that really inspired me about Steve is that he has just an amazing sort of scientific vision and this sort of ability to follow the biology and the observations wherever it led. And that's a skill and a ability sort of to take those risks that I think sometimes is lost on our researchers. I really come to appreciate that and sort of something that I adopted on my own lab. I also note that he's an amazing scientific communicator. And something that I really took to heart as well is trying to make sure that, you can tell a story around your research and work at any level for anyone that you might be talking to, whether they're expert or non-expert. So those are two that come to mind right off the bat of really role models and mentors that shaped, I think, me.
Oh, I love that. And I love that you brought up the importance of these scientific communication skills, because I think oftentimes they're overlooked. And just this idea that with Mullinish, you were able to really see how to build from the starting line an academic research lab, I think is a tremendous opportunity that I'm sure helped you later on. But I'd love to rewind back even further. I know you've got a very personal connection to sort of biomedical research. So can we talk a little bit about your scientific beginnings, if you will, Ryan, and where this interest began for you?
I can definitely talk about that. They certainly stretch back quite a ways. When you ask about the personal connection to biomedical research, that really stems from that I had pediatric cancer, a brain tumor specifically, when I was 2 years old. So basically from the get-go, almost my entire life, that is something that has been with me. And so, I went through a lot of treatments for that. Unfortunately, the tumor reoccurred also when I was 9 years old. So basically from, the start, from the time I was 2 years old, I was constantly sort of in and out of the hospitals, really relying on medications that were sort of chronic in the sense that they're sort of a replacement therapies for life. And so, seeing that firsthand, being in that environment of sort of medicine and science really, I think, determined what I was going to do with my life. Often became very interested in understanding, why things happen. How does that come about, right? And that sort of inspired me really to end up pursuing a career in biomedical research and now in drug discovery. And so that really got me interested. After I moved on to do college at UNC Chapel Hill, I actually worked in my pediatric endocrinologist research lab. So this is a person who basically had been taking care of me for, 18 years since the time I was 2 years old. He offered the opportunity for me to work in his lab. I started as a dishwasher, washing laboratory dishes. And then, over time got the ability to do more and more things, learning how to do molecular biology and some, basic biochemistry in his lab. That really opened my eyes. I think probably going into college at UNC, I was probably more thinking about medicine. But after getting experience and exposure to research in his lab, that really changed me. And, I worked all 3.5 years that I was an undergraduate in his lab. Once I graduated, I, sort of shifted from thinking about a career in medicine to career in science. And so then, as I mentioned, I worked as a research technician in Mullinish's lab for a couple of years at UNC before feeling like I was ready to move on to graduate school. And I did that. I moved to graduate school at UT Southwestern. Amazing place to be a student, to do science. I got the fortunate opportunity to join the lab of Hongtao Yu. I was in the Department of Pharmacology there, where I studied a class of proteins that are post-translational modifiers called simulation and ubiquitination, E3 ligases, the E3 ubiquitin ligases, and characterize sort of these novel proteins and how they control DNA damage response and homologous recombination. And after 4.5 years as a student there, I was in a unique position that I got the opportunity to sort of skip the postdoc and start my group as a junior faculty member in the Department of Biochemistry there at UT Southwestern. And I stayed in that position for a few years. This is a sort of unique independent fellowship position where you have funding to hire a couple of people and you sort of do your own thing and you sort of sink or swim. It was an amazing opportunity to do that. That really laid the foundation for the research I did for the following 10 years, where we characterized a novel family of proteins called melanoma antigen genes to understand that their function as E3 ubiquitin ligases. And that sort of laid the foundation for me to join the tenure track at UT Southwestern in the Department of Physiology, Pharmacology, and also in biochemistry, and continue to do amazing research there, interact and train wonderful early career scientists. And after a total of about 10 or more years in Dallas, decided to move to St. Jude Children's Research Hospital. That was a tough decision, but one that was, largely driven by my spouse was looking for a position. And obviously I listened and decided to move from UT Southwestern to St. Jude, my lab. That was a wonderful place also, especially, it was such a focus on pediatric cancer. It was amazing for me to work in that environment with my history, and just an amazing place also to do science. Then after about 5 years at St. Jude, my sort of career path took a hard left turn that I never really imagined what I was going to do then. And that was to make the move to join industry and to join Amgen, where I am today. And that was an opportunity that sort of came out of the blue that I didn't expect. The head of research here, Ray Deshays, was a colleague and a friend of mine from the ubiquitin field we both worked in. He had moved to Amgen from Caltech a couple years earlier, and he reached out to me and was like, we're going to build this new function around this concept of proximity-based therapeutics or multi-specifics, and we're looking for someone to lead it. Do you know anybody? And I responded almost immediately. I was like, Ray, I know nobody in this field. Like, I'm not an industry person. Like, I had almost zero contact with the industry prior to that.
Mm-hmm.
Then it was immediately made clear to me that he was actually asking whether I would do this job.
Oh, wow.
So then I, gave that some consideration, and then made the decision along with my family that this was something that was very unique. It was a very unique time for the science. The science that I was doing and that field in general was really transforming from sort of basic understanding of mechanisms and how things work to now to application. And this application of this concept of induced proximity, where we can control proteins in the cell through proximity to effectors, and this utilization of small molecules, in this case, multi-specifics, was sort of this concept of sort of a new drug modality. And that kind of thing doesn't come along very often. And the fact that the science was moving in this direction, that there was a real commitment from Amgen to do this, and that it was something that was not like turn the crank. You needed to be innovative. You need to be creative to do this successfully because it was so cutting edge. For those reasons, I made the decision to move to Amgen back in early 2020. And then for the next 3.5 years, it's been a whirlwind, amazing to work at a place like Amgen where the opportunities are immense and just outstanding colleagues. We're super high quality science and just grateful for the opportunity. So yeah, that's a long-winded answer, but that's how I started my interest in science and then progressed over the years through the various stages.
Absolutely. I really appreciate you sharing your path because I think it illustrates that a lot of times scientists don't have necessarily this linear career path where they knew exactly what they wanted to do. When they started the adventure, you had sort of considered medicine and then decided on research and then were in academia and then switched to industry. And I think that's very normal in science careers, but people don't necessarily realize that.
Yeah, I think that is true, that when you talk to people when they're just starting out, they always sort of imagine that there's a straight line. But that's, like you say, almost never the case. And I think it's true more and more today because there's so many different career paths open to early career scientists. I think it used to be a little bit more that everyone was thinking about sort of following in the footsteps of the mentor and running an academic lab. But I think nowadays it's more accepted and widely available that there's many other opportunities out there. And so I think it's true even more today than it used to be that the career paths are going to be winding and not straight and narrow.
Absolutely. And just kind of being open-minded and ready to take advantage of opportunities that may pop up unexpectedly, I think is really important and sort of goes back to that quote that you mentioned previously in our conversation. And I know you're doing some really phenomenal work there at Amgen, and I'd love to dig into some of the details if we can. So, Ryan, do you have a particular project that you have in the works at the moment that you are just the most excited about and want to tell us more about today?
Yeah, sure. What my group does here, as I started to allude to a little bit earlier, is we think about ways to get at difficult to drug targets. You may be surprised that more than 80% of the proteins encoded within the human genome are considered to be undruggable or challenging to drug. That means, 16,000 genes roughly for the longest time have been sort of off the table. That's a real problem. I mean, there's not that many amazing targets that are driving diseases. And if you say that 80% of them we can't touch by conventional methodologies and a way of doing drug discovery, then that's going to be a major limitation. So my group is all about building new types of technologies to reach these sort of high-hanging fruits. All the easy stuff, the sort of things that fit in the 20%, if you will, those have been picked over the last 20 or 30 years. And so we need to build new ladders to reach that sort of high-hanging fruit. And so that's what my group is all about. And so how do we do that? We do that through this concept of proximity, induced proximity. And that is taking advantage of a cell's natural mechanisms. So I said that, of course, that 80% of proteins, for example, are sort of challenging to target and regulate by conventional drug mechanisms. But nature regulates them all. So if nature can do that, why can't we sort of figure out how to control sort of those natural mechanisms and redirect them to allow us to drug these sort of difficult drug targets? And so that's where this concept of proximity-based therapeutics or multi-specifics comes in. And that is, if we can now use molecules not to do sort of all the work, meaning they don't have to sort of fit in a lock and key mechanism where the molecule binding to the target inhibits activity just because the molecule is there. Rather, if we can use sort of molecules to induce the proximity where they may bind to a target, that binding may do nothing in and of itself, but rather it induces the recruitment of other effectors, sort of natural mechanisms in the cell to come in proximity and then exert their sort of effect on that target that leads to some type of desired outcome. So a good example of one of the opportunities in this area is around inducing the degradation of these disease targets, the targeted protein degradation, where in this case you have molecules that we oftentimes refer to as molecular glues that recruit E3 ubiquitin ligases to those targets, allowing the ligase to modify the target with a signal called ubiquitin that the cell recognizes and degrades that target. So now you have this opportunity that targets that you may not be able to get to by conventional means, now you can actually get to them through this concept of proximity. And degradation is an obvious one, but you can imagine other types of proximity events in the cell or even outside the cell that will unlock more targets and allow us to develop new therapeutics to patients in need. So that's really, really what our group is focused on, and the thing that's got me really excited, and sort of this concept of any target every time, where we try to develop approaches where no matter what the disease driver is, we need to be able to get at it and drug it so that we can help patients.
I love that. And I think perhaps that sort of answers my next question, which was, you had mentioned, more than 80% of these proteins encoded are undruggable. Do you think this approach can tackle that full 80%, or are there sort of conditions in which it wouldn't be a good fit?
Well, I think everything will have limitations. I talked about sort of say protein degradation, right? Mm-hmm. That will not fit every target. So let me give you a couple of examples. For protein degradation to work, you have to have access to the sort of degradation machinery, like the ligases or the major degradation machinery in the cell, the proteasome. Well, proteins that might be like secreted outside the cell, or maybe they're in some organelle that doesn't have access to those types of machinery, like, say, the mitochondria. Those proteins are probably not going to be accessible by a classical sort of targeted protein degradation, multi-specific molecule that induces proximity between a ligase and a target, just simply because there's no ligase and no proteasome around induced proximity to. So those things will have to use other sort of proximity mechanisms beyond ligase and targeted protein degradation. And for those cases, we're developing approaches to do that as well. These are much more cutting edge. But for example, I'll give you another case of how we might get at those, is that if we can't target at the protein level, maybe we can target at the RNA level. Because obviously proteins are made from messenger RNA. And if we can control the messenger RNA abundance, we can control the protein abundance. And so we can utilize the same kind of approach I described for targeted protein degradation, but now we can use targeted RNA degradation, where instead of recruiting a ubiquitin ligase to a protein, now we're recruiting a RNase or other RNA modifying enzymes to the RNA to control its stability. So this is sort of a good example of, there's not a one size fits all. And so that's why we need multiple opportunities, and why this concept of proximity-based therapeutics has amazing potential to allow us to get at a much broader swath of targets.
Oh, that makes sense, Ryan. And I agree that this approach is just so cool. And you can just think about the tremendous potential that it could have in the medical field and just in science in general. But you alluded to, of course, there are limitations, there are challenges, and of course, there's always bad news in science, right? So I think every scientist has days where it seems like everything's going wrong. So Ryan, do you have an example of something that was a really big struggle for you, or maybe a major failure that you had to work through? And if so, could you tell me and our listeners a little bit about it? Because I think people don't often get to see sort of the struggle and the failure that comes before scientific success.
There's a lot of struggle and failure in science. And I think people go through that in different ways, right? I mean, it could be that there's a key sort of hypothesis that you're trying to test, and, you need to generate some reagents, or you need to generate some new technology to do it. And so you have to invest a lot of time to do that. And there's a lot of failures and headaches and obstacles to get through. And, I trained a lot of graduate students in my academic days. And, I feel like that's almost defines maybe years 3 and 4 of a PhD. It's those middle years that oftentimes the big struggle.
Right.
But if I think about for myself, one thing that comes immediately to mind of it was just a huge failure that was so disappointing. It actually goes way back to when I was working as a research technician before actually starting graduate school, where I just made this silly mistake, but the mistake had compromised such a investment of time in a process. So we were studying primary neurons, and we had to collect these. And it's a fairly painstaking process to go through and dissect out a very small ganglia. It's like a multi-day long process to do this. And then, of course, you have to then culture the samples for weeks on end and baby them through the primary culture, do your various treatments, and then you can collect the samples and actually collect the data, right? And so we had this sort of ambitious goal to do a biochemistry experiment, which, of course, for your listeners will know that, like, typically you need a fair amount of sample to do sort of biochemistry activity-based assays. So that's pretty hard when you talk about primary neural cultures where you have very little cells. So I was like super ambitious, right? I just started in the lab, and it's like I had this opportunity to collect these ganglia. And this was really exciting because I was going to do this, and it was going to just be an amazing set of data. And then in the end, I got through the entire process, collected the samples, ran the experiment, and the readout was like a Western blot. And I don't know how familiar you are with Western blots, but after you run like a SDS-based gel, you got to transfer the sample from the gel to a membrane to be able to probe it. And when you set that up, you have to set it up so that the sample transfers from one direction from the gel onto the membrane using a different charge setup so that it goes from one side to the other side. And if you don't set that up in the right way, the proteins, instead of going from the gel to the membrane, they go from the gel into like the buffer. And I flipped around the setup, and this weeks-long process just like evaporated right in front of my eyes.
No.
Because as soon as you open the little cassette, you can see that it transferred the wrong way. Oh, it was so disappointing. But anyway, I got through it in the end. And, this is just a prototypical sort of thing that people, whether you're new at the job, which I was fairly new at the time, or you've been doing it for a while, like, these kinds of mistakes happen, and failures happen, hypotheses are wrong, technical issues. It's all part of the job, and you sort of learn to deal with it.
Absolutely. And I think, Ryan, you can laugh at it now years later, but in that moment, when you're an early career scientist, this is one of the first projects you've ever done. How did you, react? How did you break the news to your advisor? How did you sort of like move on from that moment where you feel like you really blew it?
I had a great advisor, as I already mentioned there, and they were very supportive. I think that's important, that you have people around you that you can sort of lean on that, have gone through the same process. I was very fortunate that I also have a spouse that is in science. And so, you can commiserate together sometimes when these kinds of things happen. And Yeah, I think just having people around that you can go to that sort of understand the pain.
Absolutely. I think building that community is really important in science, not only for getting through the failures, but for celebrating the successes as well. And I do want to make sure we have a chance to talk about some of your scientific successes. So, Ryan, do you have a favorite story of a win in science that you can share with us?
You have to really celebrate the successes when they come, because as we just talked about, there are a lot of failures in scientific research. I'm not sure I would point to a particular success. I guess maybe I'll comment a little bit more broadly, and that is the thing that, over the last 10 or 15 years that really gets me excited is really seeing the success of the trainees. So whether it be graduate students, postdocs, or other staff scientists that are obviously working extremely hard in the lab to make these discoveries. And as we talked about, oftentimes they're going through the process of failures, et cetera. But when someone has really worked, so hard to test a hypothesis and that leads to some type of breakthrough discovery, that's what gets me really excited to see and gratifying to watch that process, because it's something that, like I said, doesn't come around that often. And these are the times that I feel are the most rewarding as a scientist, to be honest, seeing the success of more early career scientists.
Absolutely. I think that sort of feeling of contributing to the next generation of scientists is super rewarding. And I know, Ryan, that you actually lead the postdoc program there at Amgen. So can you talk a little bit about your role there and sort of what it's like being able to be part of the postdoc training program there?
Yeah, I'd love to talk about that a little bit. So, I've been leading the postdoc program here. This is a program that we've established here at Amgen, sort of to train the next generation of scientists. We bring in postdocs to Amgen to allow them to really do cutting edge research, get to understand the sort of drug discovery process, how industry works, and really train them for getting that next job. And most of our trainees do end up staying in industry. Many stay at Amgen or they go other places that they're interested in. And it's just amazing to see that group of scientists in our program with not only their growth as scientists, but the discoveries they're making, and then ultimately to see them be successful with publishing that work and then finding those next steps in their careers. You really feel like you're having a significant impact to each individual, as well as, of course, advancing the scientific research in the broader community.
Absolutely. And I think having trainees in the lab kind of brings a new energy, right? Keeps you motivated and inspired and sort of makes you want to work even harder in the lab. And I think that is awesome as well. But we love to encourage all of our listeners to take a step back, to take a break from science from time to time, and to read for fun. So we love getting book recommendations from everybody that we have on the show. So do you have a recommendation, Ryan, of a book that we should all add to our reading lists?
I'm not a big bookworm, to be honest. This is something that is sometimes hard to find the time, to be honest, for recreational reading. But the first things that come to mind are, I just think about the most recent things that I was reading. And the first one actually is a little bit of a funny one, and that is tend to do that oftentimes with the kids. And my 8-year-old daughter just finished reading the Harry Potter series, which was really fun to reread together with her. On a more sciencey note, I really enjoyed The Emperor of All Maladies. That was something I really enjoyed, the sort of history of sort of science and medicine. And, I think that was a good example of that. There's a few other books around that as well, but those are the two first things that come to mind.
I like it. Well, Ryan, we will add these recommendations to our website for our listeners to find. And we've talked about a lot of different aspects of your career and the work that you do. But one thing we haven't yet touched on is travel. And I think this is one of the major perks of careers in science. You do get to go to conferences, to work with collaborators and colleagues all over the world. So, Ryan, do you have a favorite place that your science has taken you?
Oh, yeah. I enjoy traveling. And you're right. That is a real opportunity that's afforded to us in our career that I appreciate. I've had a lot of interesting places. I think that one or two that are probably the most notable that come to mind are trips to China. I visited Beijing, Chengdu in the central China, and then Xiamen on the southeastern coast. That trip was really amazing for many different reasons, seeing the culture and the differences, experiencing the food and etc. But it also was really unique. I got the opportunity to go with my wife as well. So that was really fun to go together, attend the conferences, give a couple talks at different universities, etc. So that was really fun. And then just this past September, we went to Croatia also to a conference. That was an amazing place to visit as well. So lots of travel that has been memorable. Those two probably stand out more recently.
Phenomenal. So if our listeners are able to travel to either China or to Croatia in the near future, were there sites that you saw or places that you went that you think they should absolutely put on the itinerary?
Oh, yeah. I mean, in China, obviously in Beijing, it's sort of the forbidden city, sort of that more traditional historical places within China. In Chengdu, the amazing thing there was that's where a lot of the pandas are. So you can go visit the panda reserve there. In Croatia, we spent most of our time in sort of the southern part of Croatia. A really neat place there is Dubrovnik, which is a really old city, actually where they filmed the Game of Thrones. If people are a fan, it's an amazing place to visit.
Oh, awesome. Well, listeners, definitely put these on your itinerary if you have upcoming travel. And I know we've talked about some of the different people that you've encountered over your career in science. And I think the people also are wonderful assets in the career. I think being able to work with such amazing people really makes science fun. So I think people often have these stereotypes, though, about what scientists are like, and you get them often from the media, and they're just not correct. These very serious, very critical people working all by themselves in a laboratory, and that's just not what science is like most of the time. So do you have examples that maybe go against this stereotype, Ryan, or maybe a story of a quirky tradition or just a funny memory that you shared with colleagues that really shows this human side of science?
Yeah, I mean, I think that's important, right, is that you build a culture that allows people to have fun. What we're doing is serious and challenging. You do have to balance that out, right? And, one thing that comes to mind of a sort of a tradition that we had in my lab back at UT Southwestern in St. Jude that sort of reflects that I'll comment on, and that is whenever we published a paper, we would oftentimes draw up some type of funny graphic or image that sort of represents the findings. And then I have all those sort of printed out on my wall here in my office. So they're oftentimes sort of cartoony, or they could be scientific images. And it's pretty neat because it's like they have a specific connection, like each one has a specific connection to an individual, usually sort of the trainee that was doing that work. And so it reminds me of those folks. So that was one of our traditions. We also used to have annual lab retreats, and those were always fun to get to know people. And, oftentimes we would play a lot of board games, and those can get sort of competitive. And that was always fun too.
I love it. I love the idea of competitive board games. And you mentioned you're a sports person, so you are sort of competitive by nature.
I think so.
Was there a board game that you were maybe sort of like the Monopoly shark, or did you have a favorite?
Oh, favorite board game? I really like Settlers of Catan.
That's a good one.
Strategy type game.
Well, it sounds like you've had a lot of fun in your different laboratories that you've worked in over the years. And I think this is really important, kind of building that community, establishing that support system as well, because you are tackling some big challenges in science. We mentioned some of the limitations that you face or some of the challenges that you encounter. But if we took away all the barriers today, Ryan, if funding, staff, technology, feasibility, all these things that normally hold you back were not an issue, what is the one research question that you would want to answer most?
It's not exactly my area of research, but I will say that I think one of the biggest uncharted frontiers here in human biology is still understanding how the brain works. We've learned a lot of sort of at the molecular level, but where there still, of course, are mysteries to be uncovered. But understanding sort of the architecture and the systems makeup of the brain and how that forms the basis of memory is something that feels like it remains to be solved. And that's super important for not only understanding how our body works, but I'm sure will be super important and impactful to do sort of meaningful therapeutic discovery to many of the numerous neurological conditions that still remain difficult to treat. So to me, I feel like that's a frontier that still there's wide open spaces for discovery.
Absolutely. Well, as a PhD trained neuroscientist myself, you've got my vote here in terms of where those unlimited resources should go. So I love it.
Good.
Thanks for giving us something big to ponder today, just how all these different pieces come together within the brain to make us human. I think that's an amazing question. And for our listeners out there who might be on their own scientific journeys and might be encountering some of these challenges we alluded to, do you have a piece of advice, Ryan, something that really helped you on your journey that someone gave you that you can pay forward and share with them today?
Yeah, I mean, I talked a little bit earlier about the advice around being prepared and taking risks that were sort of shared with me when I was just starting out as a graduate student. The other one that comes to mind, though, is I think it was actually from my dad. And that was as I was just sort of loading up to leave for college for the first time. He stressed the importance of networking, getting to know, as many people as you can, because you never know what that'll lead to. And I think that's something I have followed that advice over my career and certainly has made a significant impact. And so I guess I would stress to folks that are sort of just starting out to try to network and get to know folks in your fields or more broadly, because I think that's something that will pay off significantly over the long term.
Absolutely. I think that is outstanding advice. I think the importance of a network, it's so important in science to be reaching out to people, to be getting to know these people in your field, because they're the ones in many cases reviewing your grants, reviewing your papers. I think having people understand who you are, what you do, and that you're good at what you do is so critical. And I think people don't necessarily realize that in science.
I completely agree. Like, this will factor into so much of everything you do. So it's really important to get to know the folks in your field.
Absolutely. Well, wonderful advice. Is there any other last piece of advice or maybe a last note of inspiration that you would like to end our conversation on today?
Yeah, I would just say follow the science, follow the unexpected observations. Those are the things that will lead to the most impactful and transformative discoveries. The things that you least expect, those types of observations, are usually what breaks open new fields. And don't be afraid to take risks. Follow where the science takes you. That's advice I've given to my trainees, and I've tried to follow myself over the years, and that's been very successful.
Well, wonderful note to end on, Ryan. And if our listeners want to learn more about you and more about the work you're doing there at Amgen, what is the best way for them to do so?
I would encourage listeners to visit amgen.com to learn more about multi-specifics and many of the topics that I talked about today. In addition, you'll find a Undruggables podcast that Ray Deshays and I, maybe it was last year we did. And then specifically to get a hold of me, of course, you can find me on the various social media outlets, LinkedIn, or even X, Twitter, at Potts Lab. So thanks so much for your leading this discussion, Marie.
Absolutely. It was such a pleasure to have you on the show today, Ryan. We really appreciate your time.
Thank you so much. Have a good day.
Thank you. And you as well. And listeners, we hope you are having a fantastic day. And we hope you'll join us for our next episode of People Behind the Science.